Module 4. Identification and Biology of Crop Diseases. Classification of crop diseases. Diseases are categorized based on causal agents and symptoms. Fungal diseases: powdery mildew, rusts, blights, smuts, and leaf spots. Fungi are the most common pathogens and often produce spores for dispersal. Bacterial diseases: wilts, leaf spots, cankers, and galls. Bacteria generally require wounds or natural openings to infect host tissue. Viral diseases: mosaic, yellowing, stunting, and ring spots. Viruses are obligate intracellular pathogens transmitted by vectors like insects, nematodes, or mechanical contact. Nematode-associated diseases: root knot, cyst nematodes, and lesion nematodes that damage roots and predispose plants to secondary infections. Oomycetes, water molds, downy mildews, and late blight, which thrive in moist environments and produce motile zoospores. Disease symptoms and signs. You examine the distinction between symptoms, plant responses, and signs. Physical presence of the pathogen. Foliar symptoms: chlorosis, necrosis, leaf spots, wilting stem and root symptoms. Cankers, galls, rots, lesions. Fruit and seed symptoms: spots, rots, deformation, discoloration. Pathogen signs: fungal spores, bacterial ooze, nematode cysts. Pathogen biology and life cycles. Fungi: sexual and asexual reproductive cycles, spore types, and environmental triggers for germination bacteria. Binary fission, survival in soil, water, or plant debris, and infection mechanisms. Viruses: transmission by vectors, systemic movement within the plant, and replication strategies. Nematodes, egg, juvenile, and adult stages, modes of feeding, and host specificity. Infection mechanisms and disease development. Pathogens invade host plants through natural openings, wounds, or direct penetration. Environmental conditions such as temperature, humidity, rainfall, and soil pH significantly influence disease development. Module 4 explores concepts such as latent period, incubation period, secondary spread, and epidemic progression. Epidemiology and disease dynamics. You study the factors affecting disease incidence and severity, including pathogen population density, host susceptibility, environmental conditions, and management practices. Models of disease progression and forecasting tools are introduced to anticipate outbreaks. Diagnostic techniques. Accurate diagnosis is crucial for effective disease control. Techniques include visual scouting and symptom recognition, microscopic examination, culture-based isolation and identification, molecular diagnostics, PCR, ELISA, DNA barcoding, remote sensing and digital imaging. Integrated disease management principles. Building on previous modules, learners explore strategies to minimize disease impact sustainably. Cultural practices, crop rotation, sanitation, planting resistant varieties, and proper spacing. Biological controls. Use of antagonistic microorganisms, mycoparasites, and beneficial nematodes. Chemical controls. Fungicides, bactericides, nematicides, and proper application timing. Physical controls. Solarization, pruning, and removing infected plant debris. Case studies and practical applications. The module includes examples of major disease outbreaks and successful management practices in cereals, legumes, vegetables, and horticultural crops. You are encouraged to integrate theoretical knowledge with practical field-based problem solving. Learning outcomes. By the end of Module 4, you will be able to identify and classify major crop diseases based on symptoms, signs, and causal agents. Explain the biology, life cycle, and infection mechanisms of pathogens affecting crops. Analyze the influence of environmental and host factors on disease development. Apply diagnostic techniques to accurately identify crop diseases. Design and implement integrated disease management strategies for sustainable crop protection.